Assalamualaikum. My name is Krishna from the Department of Biotechnology and my role number is 009. Today the topic of my presentation is hydrogen nanoparticle techniques in drug delivery. So first of all, we should know what are hydrogen nanoparticles. Hydrogen is a network of polymer chains that are hydrophilic, sometimes found as a colloidal gel in which water is the dispersion medium. A three-dimensional solid results from the hydrophilic polymer chains being held together by crosslinks. Because of the inherent crosslinks, the structural integrity of the hydrogen network does not dissolve from the high concentration of water. Hydrogels are highly absorbent. They can contain over 90% water, natural or synthetic polymeric network. The second thing which we are going to discuss is chemistry of hydrogen nanoparticles. The crosslinks which bond the polymers of a hydrogel fall into two general categories, physical and chemical. Physical crosslinks consist of hydrogen bonds, hydrophobic interactions, and chain entanglements. A hydrogel generated through the use of physical crosslinks is something called a reversible hydrogel. Chemical crosslinks consist of a covalent bond between polymer strands. Hydrogels generated in this manner are sometimes called permanent hydrogels. Further, we can discuss its chemistry. One most developed method of initiating a polymerization reaction involves the use of light and stimulus. In this method, photoinitiated compounds that heat from the absorption of photons are added to the precursor solution, which will become the crossing formed in the hydrogen to be controlled. The properties of a hydrogel are highly dependent on the type and quantity of its crossing, making photopolymerization a popular choice for fine-tuning hydrogels. This technique has been considered. The next thing is, what are the uses of hydrogen nanoparticles? Here we shall discuss the uses of hydrogen nanoparticles. Hydrogel-coated wells have been used for cell culture. Environmentally sensitive hydrogel, also known as smart gels or intelligent gels, these hydrogels have the ability to sense changes of pH, temperature, or concentration of metabolites and release their load as a result of such a change. Injectable hydrogels, which can be used as drug carriers for treatment of diseases or as cell carriers for regenerative purposes or tissue engineering. Sustained-release drug delivery system and strength, pH and temperature can be used as a triggering factor to control the release of the drug, providing absorption, deslogging, and debriding of necrotic and subtropic tissues. Hydrogens that are responsive to specific molecules such as glucose or antigens can be used as a biosensor as well as in PBS. Disposable diapers, whether absorbed urine or in sanitary napkins. Hydrogens are used in contact lenses, silicone hydrogels, polyacrylamides, and polymetcons. Hydrogens are also used in cell and tissue engineering applications due to their ability to inject or hold a precursor solution or use a cell into a home site, then solidify it in situ. Now we shall discuss nanotechnology-based drug delivery systems. Several anti-cancer drugs, including paclitaxel, doxorubicin, bifloralizidazole, and dexamethasone, have been successfully formulated using nanomaterials. Polylactic glycolic acid (PLGA) and polylactic acid (PLA)-based nanoparticles have been formulated to encapsulate dimethicone or a glucocorticoid with an intracellular site of action. Dimethicone is a chemotherapeutic agent that has anti-proliferative and anti-inflammatory effects. The drug binds to cytoplasmic receptors, and the subsequent drug-receptor complex is transported to the nucleus, resulting in the expression of certain genes that control cell proliferation. These drug-loaded nanoparticle formulations that release high doses of drug for prolonged periods of time completely inhibit proliferation of vascular smooth muscle cells. Colloidal drug delivery modalities such as liposomes, stem cells, or nanoparticles have been intensively investigated for their use in cancer therapy. The effectiveness of drug delivery systems can be attributed to the small size, reduced drug toxicity, controlled delivery of the drug, and modification of drug pharmacokinetics and biological distribution. Too often, chemotherapy fails to cure cancer because tumors are resistant to multiple anti-cancer drugs. In most cases, the resistance develops when cancer cells begin expressing a protein known as P-glycoprotein that is capable of pumping anti-cancer drugs out of a cell as quickly as goes through the cell's outer membrane. New research shows that the nanoparticles may be able to get anti-cancer drugs into cells without triggering the P-glycoprotein pump. New research shows nanoparticles may be able to get anti-cancer drugs into cells without triggering the P-glycoprotein pump, as I discussed earlier. The researchers studied in vivo efficacy of paclitaxel to add nanoparticles in paclitaxel-resistant human colorectal tumors. Paclitaxel entrapped in emulsifying wax nanoparticles was shown to overcome drug resistance in a human colon adenocarcinoma cell line. The insolubility problems encountered with paclitaxel can be overcome by conjugating this drug with albumin and paclitaxel bound to biocompatible proteins like albumin and vaccine, the injectable nanosuspension approved for the treatment of breast cancer. Solvent Creamer-4-EL means increased formulation of paclitaxel causes acute hypersensitivity reactions, may reduce the risk of allergic reactions when giving paclitaxel. Patients must undergo pre-medication using steroids and antihistamines and given the drug using slow infusions lasting a few hours. Now we are going to discuss what are nano drugs or what nano drugs actually are. Pharmaceutical companies do not expect nanostructured materials to become new drug compounds. However, carbon balls and nanotubes might be useful in drug delivery vehicles because their nanometer size enables them to move easily inside the body. The active compounds might be inserted in a nanotube or bonded to particle surface. Other types of nanopowders or biomolecules are also useful and are closer to market space. Cosmetics based on quantum dots are already sold in large quantities. Nano-based technologies use nanocrystalline particles such as zinc oxide. For users from screens and other products, the particles are pretty active and cause minimal damage to DNA in small light. Quantum dots are manufactured how? Quantum dots are manufactured between 3 and 5 nanometers, suitable for binding specific biomolecules. The quantum dots are luminescent particles more stable than organic dyes used today, and the most important thing is that they are not toxic. So winding up my discussion on hydrogen nanoparticles, I would like to say that nanoparticles are promising for delivering compounds to improve the pharmacological and therapeutic properties of conventional drugs. Incorporating drug molecules in nanotherapies provide molecular advantages like better biodistribution of compounds, protection against degradation, improved drug attachment, passage targeting, exposure, and communication with biological barriers. It is also nanoparticles mediated drug delivery systems will revolutionize and be very useful for the care of mankind. And that's all from my side. Thank you.